Hey everyone, this is Mr. Jennings here. Today we are going to go through meiosis, karyotypes first for a minute, just to kind of review. This is actually something we covered in class a couple weeks ago. Just want to have a nice little recap here, just refresh your memory. Because it's been a couple weeks and just want to make sure we have it. The notes that we're going to have in this next set of videos for this week, you already have a printout of. I gave you all a printout of these notes, I believe, before. You should have them somewhere in your belongings. If not, obviously I can send the PDF, the copy of the PowerPoint up when it's available. Okay, so we're going to go into it right now. So today we are again going to talk about karyotypes and chromosomes and how we track chromosomal disorders. Let's go there. Okay, so each of the human chromosomes will look like we see here. Remember we did this chromosome activity where you had to match off chromosomes on computer. If you did not do that, you could always go back and work on that just a little bit. Don't know when we can be able to turn that in if you did not get to. Now, each chromosome here is ranged based on size. Now, when we do this size arrangement, this is going to become what we call a karyotype. This is a photomicrograph. Basically, they take a picture of a microscope's image and arrange them according to standard classification. Typically, the longest is number 1, with the shortest being chromosome 22. And X and Y being their own little subgroup, being the sex chromosomes. So chromosomes are digitally arranged so that they are matched with their homologous or partner chromosome. Basically, everybody has 2 copies of each chromosome, one from your mother, one from your father. We talked about that in last week's video. That's that law of independent assortment. Homologous chromosomes are the same size, shape, and they carry the same genes for a particular trait, but they don't carry the same version of that trait. They could have different alleles. And again, they are numbered according to size. Again, each homologous pair, they code for the same trait, but the genes can have different versions of different versions of the alleles. So, for instance, one chromosome could have the alleles for red hair, the other chromosome could have the alleles for blonde hair. Karyotypes is also used in sex determination. We usually look at this karyotype here, for instance, that we see on the screen, have 23 exact pairs. That means the person is female because the X chromosomes are identical. So chromosome 23, which is actually not numbered as 23 on this image, are both X. Males, the 23rd set of chromosomes, again, not numbered actually 23, they have X and Y. That means heterozygous for the chromosome. Now, if you look at this picture here, the question is this person female or male? As we look, you may notice down here, there's 2 X's and no Y's. That's how you know for a fact that this one here, based on the chromosomes, is female. So there are ways we can use karyotypes for identifying various genetic disorders. Chromosome, for instance, Trisomy 21 is an abnormality shown in this karyotype here because they have an extra copy of the 21st chromosome. Let's see all the way down here. That person is going to be classified as somebody with Down syndrome, and that's based on the 23rd chromosome. This is from the National Down Syndrome Society. Down syndrome is a spectral disorder. Basically, anybody who has the 3rd copy of the 21st chromosome has a variety of different phenotypes because there's multiple ways for it to be expressed. Having extra copies of chromosomes in most eukaryotic, especially vertebrates, tends to be challenging for vertebrates because of how tightly organized our systems are. But the 21st chromosome has pretty low impact, so usually trisomy 21 results in, viable offspring. But other chromosomes may not have as much of a reduced impact. For instance, if you have trisomy 1, it'd be too great that usually the child does not make it to term. Chromosome counts matter for a variety of things. Conversely, plants, for instance, another eukaryotic organism, them possessing an extra chromosome is actually usually viewed as beneficial, and most plants in the horticulture department have multiple copies of each chromosome. They display polyploidy. There's also a correlation between trisomy 21 and the age of the mother. Typically, the older the mother is, the higher the chances of them having a child with Down syndrome. So whenever you are expecting, one of the things they ask you is for your age. They do a little survey, a little questionnaire with the mother and the father if possible, with the doctor, and the doctor's going to ask you a bunch of questions. So they did this with me and my wife when we were having our sons. And they ask a bunch of questions about family history, and they ask about age, because age is a reliable indicator for the frequency of trisomy 21 incidence. This comes down to just an event called non-disjunction, and it's more prevalent in women who are older, because the regulations for non-disjunction tend to reduce. Actually, most of our body's internal regulations decline as we get older, and that's part of the aging process. Other non-disjunction disorders, Meiosis X, this is abnormally shown karyotype here where they only have one X chromosome. This person has what we call Turner syndrome. Functional, for the most part, fully functional female. There's some differences, and we're going to do a chromosome disorder project this week, and you can look this up on your own if you're interested. XXY male, having an extra X, is typically called Klinefelter syndrome. Biologically speaking, they are recognized as male, but they'll have more feminine characteristics. Now, the way they get these DNA samples is a variety of different ways. Typically, they do what's called amniocentesis. Basically, they'll draw out amniotic fluid from the placenta with cells containing the fetus. They don't do this for every female. For every pregnancy, they only do this for certain pregnancies that are considered high risk for certain genetic disorders, or if you opt into it. We were given the option to opt into it, me and my wife. We decided not to because we knew the risk was low, and it's a somewhat invasive procedure, so you don't want to risk unnecessary complications. But what they do is they take the cervical fluid, and then they'll analyze the cells and look for cells undergoing the baby's DNA, baby cells that are undergoing mitosis, and they'll find prophase, and they'll take a picture of it and do a karyotype. Another way of doing it is Chorionic villi sampling. Basically, they'll take, instead of going for the amniotic fluid, they'll take cells from the chorion with fetal tissue. It's actually not the child. The fetal tissue is on the outside of the placenta, but they could do that as well. Now, if the chromosomes are if there are chromosomal numbers abnormalities, how do they form? It's through meiosis, and we're going to do another video here reviewing meiosis because it's been a while. But meiosis is the process of creating sperm and egg from a diploid cell. If there's a mistake when chromosomes are separating, then the resulting sperm and egg cell either have too many or too few, and those mistakes are called non-disjunctions. I think I'm going to pause here. Will be about meiosis. I'm not going to go on to it just yet. I just want to make sure we kind of go through it right now. So, non-disjunction events occur naturally. They're a normal process of development. In some organisms, like plants, non-disjunctions are actually viewed positively. And in other organisms, like vertebrates, it tends to be difficult because the body is tightly organized and tightly regulated. Sometimes having extra chromosomes or proteins, and that creates a complexity. Next time, though, we're going to talk about how this actually occurs. We're going to talk about meiosis. It's just kind of a review. It's going to be how sperm and egg cells are produced, because we started talking about this well back in March, back in February a little bit, and we're going to make sure we kind of review and recap on that. Thank you for watching. Thank you for listening. Keep watching for more. Have a great day.